It's an honor and privilege to have one of my old colleagues back from Baltimore, Dr. Reza Shadmehr. I don't think Dr. Shadmehr needs any introduction, but I will just do it. He is the professor of biomedical engineering and neuroscience at the Johns Hopkins University, and his areas of interest have been traditionally motor control and limb motor control, reaching, and also to some extent eye movements. So I'm pretty sure that you guys will all enjoy his talk, and you will learn a lot, and it'll be fun. Thank you.
Well, hi everyone. Asif, thank you so much. I want to first tell you what a privilege it is to be here. Many years ago, perhaps 25 years ago, I was visiting, and I learned about FES. I learned about how control of shoulders can be used as a way to close the grip of the patient using functional electrical stimulation. And for the next 25 years, I taught that material, the videos that I borrowed from Bob, in the medical school courses that I teach. And so it was a big influence on me. And it's really lovely to see how the work has progressed. Now the cortical stimulation, now the cortical decoding activity that involves the muscles of the arm. It's really come along. And it's a testament to you as a leader and to you guys as the graduate students and the faculty of this great department. So congratulations on that. My own path has moved to animal work, and particularly non-human primates, because I believe that in order for us to understand the mechanisms by which the structure that I'm going to tell you about, the cerebellum, controls movements, it's been useful to try to get closer to the neural basis of it. So I want to begin with telling you about my students who have been critical for this. These are all PhD students that have graduated. Ehsan, Paul is a current student, Jay is a current student, Amin is a current student, and Alden is a current student in my lab. PhD students whose work I'm going to be highlighting today in my lecture. So I'm going to be talking about the cerebellum. And in order to get us started, I want to give you a little bit of background about what do we know about this structure. Much of what we know began in the early part of the 20th century because of World War I. And this is what I learned from a close colleague of ours, David Z. Basically, as the British soldiers were involved in battle, they suffered gunshot wounds to the back of the head because of this poor helmet design that you see here, whereas the Germans had this helmet that, covered their whole head. So there was this neurologist in England, his name was Gordon Holmes, that examined these patients when they returned from battle, and he highlighted two basic problems that he saw. One of them you're familiar with, it's called dysmetria. Basically, the individuals that had cerebellar damage would make movements, but then they had these errors at the end of their movements. And the other problem is called hypotonia, which was basically it's hard for them to maintain their limb at a particular position. So it tells you that cerebellum has something to do with control of movements and holding still, control of posture. The second major idea comes from computational work that started with David Marr and James Albus, and then biological work that Masao Ito did. This has to do with learning. This idea is that there is a teacher for the cerebellum. The cerebellum is a learning machine. There's this teacher here, is inferior olive, something that gives rise to what's called climbing fibers. And then it's the teacher of the students of the Purkinje cells of the cerebellum. So the cerebellum is a learning machine. It has a teacher. The teacher is the inferior olive. The third major idea came about 30 years later, and it's from Chris Mile and Daniel Wolpert. This is also a computational idea. It's this concept of forward models. The idea that what the cerebellum is learning is to predict the consequences of actions. So somehow when you do things, the sensory things that you expect to get is being predicted by the cerebellum. This is two computational ideas, learning and forward models. One, a clinical idea, has to do with the deficits that happen when there's damage to the cerebellum. And as I present, please raise your hand if you have questions. I'd be happy to answer your questions for you. So this is Maurice Smith. Asif was asking me about Maurice. Maurice was my second graduate student. He's now a successful professor at Harvard. He's doing very well. He's holding this thing that's called a manipulandum back then, and he's making reaching movements, and you can see he's doing it just fine. If you take a cerebellar patient and you have them do the same movements, you see they have these troubles at the end of the movement. I'm going to focus on saccades, and here is a patient that's going to make saccades for you. Let's see if we can do it.
So Reza, this guy's from Cleveland.
Yeah, is that right? You know him?
He's one of our patients.
Oh my goodness. Okay, it's working on it. I think it's going to show it to you. Let's see if it succeeds. Okay, it isn't going to show it to you. All right. Well, what you would have seen, it's wonderful that to learn that he was from Cleveland.
Yeah,
I didn't know
he
that.
had scalzi. He's one of John Lee's patients.
Yeah.
And they inherited the whole family.
That's amazing. So basically, if you were to see him making these saccades, what you would see is that the eye would overshoot, the eye would overshoot, the eye would overshoot, and finally it would land on target. And you can imagine how debilitating life would be if every time you were trying to read a sentence, the eyes would need to do so much work for you in order for you to foveate the target. So you see that the movement starts out okay, but it doesn't land on target. And so this raises the question, so what is the cerebellum doing that's so essential for these ending of these movements? All right, so I'm going to focus on saccades, this perhaps the most elementary voluntary movement that we make. In order to do that, I want to teach you a little bit about the anatomy of our circuit. Now, from your retina, you have direct projections to a region in your brain called the superior colliculus that has access to the machinery that would produce this goal-directed movement. But you are not a frog. You are not allowed for your colliculus to make the decision of move your eye. Rather, that same input goes to your cortex. You evaluate the scene around you, and you direct your gaze toward things that you value. The basal ganglia removes the inhibition. The frontal eye field and the lateral interparietal sulcus produces excitation to the location where there's something of value. That is the instruction that the colliculus needs. Now you generate an eye movement. The eye moves to the location where you're interested. As you do that, there is a copy of these commands that goes to the cerebellum, and the cerebellum does some kind of computation, some kind of magic that becomes this prediction that makes it so that this circuit can produce the movement that it needs to, and your eye lands on target. So if you look at this circuit, what you will see actually is the principles of neuroscience. If you pick up that book, you will see the names of these individuals that have contributed to this very simple movement that I showed you. So this is really a fundamental process that the brain is doing, and we know a lot about it. So you might ask yourself, okay, Reza, who the heck are you to tell us anything new about this simple movement like a saccade? And what I'm going to show you is that the puzzle is here, is this structure here. Because when you look at this structure, what you see is that we can see that in these patients that there's these deficits in these movements at the end of it. But when we look at the neural activity, we just have a really hard time understanding how could that deficit come about given the neural activity that you see in the cerebellum. What's the relationship between the neural activity and the deficit that exists when there's damage to the cerebellum? And now, why is it important to know that? At the end of my talk, what I'm going to show you is that understanding how a circuit works might be really critical to then overcoming the deficits that are associated with diseases of that structure. That's the goal. Okay, so this is the model system that we work with. This is called a marmoset. It's a New World monkey. This is the cerebellum of this particular individual, this marmoset. And the part of the cerebellum I'm going to be talking about is this lobule 6 and 7 of the vermis called the oculomotor vermis, the region that's important for control of this movement. Now you might ask yourself, Reza, why the heck are you working on this? One, it's a primate, which means that it makes saccades. It has a fovea like you and me, and it's also a beautiful animal. I think it's worthwhile working with beautiful things. Second, it's small, which means that many of the tools that were developed for rodents can be applied to this primate. So what happened to me was that an idea came about describing how the process of control might be happening, and I wanted a way to test it, and we decided to work on marmosets because of the advantages that I felt it had. And I think it's the future of non-human primate research. All right, so what do we see? Let's look at some movements of this particular animal. So these are saccades. Here's the saccade to the left. This is the velocity of the eye. This is the velocity of the eye to the right. Here's a Purkinje cell, a principal cell in the cerebellum. And you see in this case, it's pausing. And now what you notice is that there's really nothing special happening, at the end of these movements. It's just pausing. Here's another cell. Here's a third cell. Here's one that's bursting. Here's one that's bursting and pausing, and so forth. So the activity of these cells has puzzled neurophysiologists because when you look at this activity, it's just hard to make sense of the relationship between this activity and the behavior that you're looking at. So that's the problem of decoding. How do we go from neural activity to understanding representation and control? This is the puzzle that I want to talk with you today. All right. What's the idea, Reza? Well, the idea is as follows. What David Marr and Masao Ito talked about is to view the cerebellum as a learning machine. The cerebellum is basically a three-layer network. It gets inputs as mossy fibers, granule cells, Purkinje cells, nucleus cells. That's the output. If you want to train a three-layer network, what you do is that you compare predictions with observations. That becomes the error. That error is the teacher of this network. It's actually quite interesting that in the cerebellum there's something similar. You also have this input that is what we think is prediction errors. And that was Masao Ito and David Marr's idea. So, all right, here are Purkinje cells. The principal cells that receive this prediction error, it's rather weak input to the nucleus, but extremely strong to the Purkinje cells. And I hope this plays for you so we can see what it looks like. All right, so it doesn't have sound, unfortunately. If you could hear it, what you would hear are these spikes down here, which is the simple spikes of these Purkinje cells. Those are the predictions that it's making. And these spikes that you see here, they're like thunder, like this in a neurophysiology lab. You hear this thunderous thing that comes back, and that's the complex spike. That's the information that's coming from the inferior olive, presumably saying something about the error that's being sent back. So what's special here is that using this neurophysiology, you can first identify this is a Purkinje cell, and second, you can dissociate its outputs, those simple spikes that it's making, from the error feedback that it's getting from the inferior olive. All right, so what's the idea of putting together this concept, being able to decode this activity? And the idea is like this. Imagine you have a schoolroom where you have a teacher. We're going to have a whole school made up of a bunch of school rooms where you have teachers in each room. The teacher is the inferior olive. We're going to label Purkinje cells based on who's their teacher. By identifying their teacher, we're going to build populations. And the idea is, if you have a teacher, you're going to learn differently than if you have a different teacher. And by identifying who's your teacher, we're going to build populations. So it's an anatomical hypothesis that we're going to use to describe computation. This is the work of my student David Hertzfeld, and it goes like this. A population of Purkinje cell converges onto a nucleus cell. That's what makes a prediction. There is this observation that comes. You compare the two. That's the error. That error is sent to these students here that then is going to produce their output. So to organize the Purkinje cells, what we're going to do is to organize them into populations that care about the same error. This is the main idea. This is the idea that led to basically me building a monkey lab. And it goes like this. To understand the language of the simple spikes, I think it's going to be useful to organize the cells into populations that share the same error information. Who is their teacher? Identify their teacher, label them based on their teacher, organize them into populations, then put together their spikes and see what they're saying. This is the basic idea. So we built a lab. We recorded from the neurons. And here's the first experiment that we did. We first identified what error they cared about. So that's complex spikes. So here's what the monkey does. Sits in a dark room, you give it a fixation point, you give it a target. The monkey makes a saccade. At the end of the saccade, the target is not on the fovea, it's someplace else. You look at the response to that error. So here's when the saccade ends. These blue guys are those simple spikes. That's the predictions that the Purkinje cell is making. And this red thing, that's the complex spike. So here's the saccade ends, and you see after the saccade ends, there's some activity. This is for leftward errors. Here's for rightward errors. You see there's a little bit less activity. And you can count these responses and you get a tuning function. You see that this cell cares a lot about leftward errors. This is probability of a complex spike, not so much about rightward errors. So all right, this cell responds to leftward visual errors. All right. Now, you can look at the response as a function of time, and you can say, all right, in the direction that it prefers the error, here's the response. In the opposite direction, 180 degrees away, you see there's a suppression. So we get the concept now of a coordinate system. This is step 2. How do we use this information? We use the preferred direction of error to define this new coordinate system. Say you have a Purkinje cell. Say this is the direction of error that it cares about. Say you make a saccade. This is what you would call a 45-degree saccade. But actually what this is for this cell is a saccade in direction preferred error plus 250 degrees. So now what we think about is, where is this error coming from? Let's see what the saccade was doing. So when you are asked to look at this dot on the screen, what happens is that there is going to be activity in the rostral pole of your colliculus. When I give you a target, there is all of a sudden an event that takes place that produces activity in the different part of the colliculus. That was unexpected. That engages the inferior olive. You get complex spikes here. Now, you decide to make a saccade. The activity in the rostral pole goes down. The activity here rises. The eye moves to the target. But the target is not on the fovea because I've moved it. It's over here. So you get a new location. This is what you expected. This is what the actual sensory feedback is. That also engages the colliculus and you get activity. So what I'm doing is that I'm recording from climbing fibers here, but engaging the colliculus. So I am mapping the superior colliculus onto the cerebellum and asking what part of this visuomotor space does this Purkinje cell care about. Okay, so what is the result that we get? So let's suppose that for you, the complex spikes have an on direction here. I present you a target. Suppose the target is in direction CS on. You see a response at about 80 milliseconds. This is a typical collicular response. And then you see a response before the saccade. Look at the y-axis here. This is going from 0 to about 1 hertz. So this error information that's going to the cerebellum is very low frequency. Move the target slightly away, you see that it produces smaller activity, visual response, motor response. A little bit away, you get a little bit less even when it's 180 degrees away. So you have a response field associated with this error. It has a preferred direction. All right. Now, the whole point of doing this was to understand those predictions that are being made by the Purkinje cells. So now let's look at their simple spikes. What we see is that during fixation, while you're holding still, the cells are suppressed. These are inhibitory neurons. They're projecting to the nucleus. While you're holding still, there's an active process because you see there's this suppression of the Purkinje cells. When a saccade takes place, you see that in direction CS plus 180, there's a burst that's early and strong. In direction CS on, there's a burst, but it's later and weaker. Okay, so we still don't really understand what this means, but we at least have this coordinate system and we see this activity in the population. So to summarize what I've seen so far, the olivary input is reporting events in a specific part of the visuomotor space of the Purkinje cell. The simple spikes are suppressed during a fixation. They don't respond to the visual event. As a population, they burst during saccades, but earlier and stronger when it's in the direction opposite to the preferred error.
Take a question.
Yeah, of course.
So are these authors give those histograms cleaner in this relative to their predicted or their orientation than if you just did them in the real field?
Yeah, absolutely, they are. But more importantly, I'm going to show you that the downstream effects of those simple spikes match with the CSR. So that's the next step. To understand what these spikes mean, it would be nice if we had a causal way to manipulate them and see what's their effect on behavior, right? Now, how would you do that? Well, you might say, well, maybe you would do optogenetics to stimulate it. But the problem with optogenetics is that you can't just stimulate the Purkinje cells that have the CS on in this direction. You get a whole bunch of them. So how are you going to make it specific to a small number? Here's what we did. This is my student Solomon Mueller. And here's the experimental idea. When a complex spike occurs, when the olive sends its error information to the Purkinje cells, for 20 to 30 milliseconds, that Purkinje cell is disabled. It's unable to send spikes. There's this period of suppression that takes place. It's such a strong input that basically the cell becomes immobile in its ability to transmit information for 20 to 30 milliseconds. So what he's done here is to align the data, and you can clearly see these black dots, which are the simple spikes, are suppressed after the complex spike arrives. All right, so if a saccade takes place and there is no complex spike, you see this bursting activity. If there is a complex spike sometime here, the cell is suppressed. What's the downstream effect of this suppression on this movement? So you had this complex spike that arrived before the movement, and it caused the suppression, or during the movement caused the suppression. What's the downstream effect? So imagine a coordinate system. So here's the horizontal velocity, here's the vertical velocity, here's the saccade to some target. It goes out in this velocity space. Suppose this was without a complex spike. Here's with a complex spike. Maybe there's some small difference between this. Now let's put a coordinate system here. Let's put the preferred error here, 90 degrees here, and let's project the change in the saccade's trajectory onto this coordinate system. What do we see? We see that if you suppress the activity of this Purkinje cell during this movement, there's no change in eye velocity along CS plus 90, but it pulls the eye in direction CS on. What this says is that suppression of that simple spike had a specific effect on behavior. It had a downstream effect that pulled the eyes in the direction of its preferred error. So it's not just some input that's coming, it's describing a whole anatomy of the projections of this Purkinje cell. What is this anatomy? It looks like this. Basically, you have a direction of CS on, you have a direction of CS plus 90. The inferior olive is telling you, I care about these errors here. And what that's doing is that downstream effect is to pull the eye in the same direction as the error itself. So this has a very fundamental thing to tell us about how to now put together the spikes. It goes like this. Suppose a saccade takes place. For some population of Purkinje cells, that's in direction CS on. For another population, that same saccade is in direction CS plus 180. For this group, the downstream effect of its simple spikes is an agonist. For this group, the downstream effect is an antagonist. Therefore, the population response is the SS rates in direction CS plus 180 minus the SS rates in CS on. Let's now go back to our simple spike activity. This is what we had. A burst that was early and strong, a burst that was later and weak. What I showed you is that the downstream effect must be the subtraction of this minus that. What does that happen? Here's what it looks like. It's an agonist burst followed by an antagonist burst. Let's line that up to peak velocity, and you see there's this beautiful alignment that shows that the antagonist activity is coming and peaking right before you're supposed to decelerate that movement. Some saccades are made when the animal is provided with reward, sometimes unrewarded. The rewarded saccades have high vigor, the unrewarded ones have low vigor. And look, for high vigor saccades, you get this beautiful increase in the antagonist activity, predicting when to stop the movement. Moreover, what we see is that the spikes align in time. So this is a measure of synchrony. One of the beautiful things about this animal is that because they're small, we can use these fabulous silicon probes and record from many Purkinje cells simultaneously. And what you see is not just that on average the firing rates are doing what I showed you, but they become synchronized in time so that you get this increased alignment of the spikes during the period when you should be decelerating. So the simple spikes show an agonist-antagonist burst, and they show this increased synchrony during saccades. This is, yes, Asif.
So low vigor and high vigor,
Yeah.
rewarded versus non-rewarded. So far we are talking about Purkinje cell, simple spike, complex spike, and the influence from the inferior olive.
Yeah.
But when it comes to reward, are we looking at basal ganglia now?
Yeah, on the colliculus, which then reflects the complex spikes activity. So basically, what we see is that the effective reward is a modulation of collicular activity that's directly potential. Well, we see it in the complex spikes. You see much greater modulation of complex spikes for high reward than low reward. Yeah, and what does that have to do with control of this movement? I don't know, but the simple spikes tell you that I better decelerate sooner and stronger if it's a faster movement.
Or it could be looking at the dollar bills, that experiment that I remember.
In humans, yes. Yes, sir.
Yeah, can you say something about the distribution and the topology? Are the cells, Purkinje
Yeah.
cells with a similar orientation,
So-
clustered together, and then would you expect a field potential out of that?
You know, unfortunately, the vermis is a midline structure, and so it's really hard to know if you're right or left of the midline. But roughly speaking, it's contralateral. So cells, Purkinje cells on where I think are the right of the midline tend to have leftward preferences for error. But it's really hard because of this mixture that takes place, because it's a continuous structure, basically. Okay, so let me summarize where we are. Purkinje cells show a preference for error, probably because they get input from a specific region of the superior colliculus. When a Purkinje cell simple spikes are suppressed, the eyes are pulled toward the direction of that preferred error. That's really a critical element of anatomy that's been discovered, well, at least predicted, by this functional result. When we organize the Purkinje cells based on the preference for error, which is really, if you think about it, is this old idea of Georgopoulos, really. You can imagine it this being preferred direction, but this is now not a direction, but a preferred error input from the olive. What we see is that the spike rates and synchrony are suppressed during fixation. The spike rates show an agonist-antagonist burst that peaks just before deceleration, and they synchronize during saccades. So the next topic I want to show you has to do with the idea of having different teachers. What if you have Purkinje cells that get input from a teacher that teaches you something about visual events and a teacher that teaches things about motor events? You would expect them to produce different kinds of activity. This is my student Jay Pai, and let's go back to the data to show you what we find. So I showed you that when the target is presented, you have some activity, and when the saccade is presented, you see some complex spikes rate increase before, and it's directional. Now, you might ask, well, are these two things the same or are they different from each other? Let's focus on a event that is like this. So we present a target to the subject, but the subject decides to make a saccade elsewhere. This is just voluntary. The animal can ignore this target and decide to saccade elsewhere. If you look at the response to the target when target was presented, this is the complex spikes now, 100 milliseconds or so after the target is presented, you see that the complex spikes are coding where the target is, not where the animal is going to make a saccade. But if you look at the moment before the saccade, you see that the complex spikes are telling you where the saccade is going, not where the target was. Okay, so you can dissociate these two things. So the inferior olive is telling the cerebellum the direction of the target, and then telling the cerebellum, here's the movement that you're about to make. The colliculus is about to make a movement someplace, and is reporting it in the stochastic, low-frequency way. All right. So it's reporting both the direction of the target and independently the direction of the planned movement. All right. Now, I'm going to show you that these two pieces of information is actually going to do two different Purkinje cell groups. And it goes like this. There's one type of Purkinje cell that receives the visual input at its 70 or 80 millisecond latency, and there's another group that doesn't get that and is active before the saccade. So let's compare these two groups and how they respond to this task. You give a target, the animal decides to saccade elsewhere. So there's that type 1, the one that has complex spike response to the target, that indeed is encoding target location, not the saccade. However, before the saccade actually takes place, it only has a weak encoding of the movement. The second type has a very strong encoding of the movement, but not the visual event. So, if you think about it, you might imagine these are the visual, perhaps in the more superficial layers of the colliculus that projects to these inferior olive neurons that go to the Purkinje cells. Perhaps these are deeper layer colliculus neurons that project onto the inferior olive that give you the actual saccade. But the bottom line is that there's one group of Purkinje cells that gets teacher information about the visual event. There's another group that gets teacher information about the motor event. Now, the hypothesis is if you have two different teachers, you're going to contribute to two different aspects of movement. Is that true? Let's now look at the simple spikes. What do we see? We see that that type 1, the one that got visual information, is suppressed during fixation. Type 2 is not suppressed during fixation. So this type cares about holding still. This type does not. So, at least from the perspective of holding still, these two groups of Purkinje cells differ. Then you look at their simple spikes during the movement, and you say, okay, there's some activity during the movement. Let's look at the difference between CS plus 180 and CS on. What do we see? Type 1 is mostly interested in providing an agonist burst. Type 2 is interested in decelerating your movement. So, Purkinje cells that receive visual information from the olive produce simple spikes that accelerate the movement toward that direction. The ones that receive motor information produce simple spikes that decelerate that same movement. So the coordinate system of this error determines the downstream effect. The type of information that you provide, visual or motor, makes me help make that movement or stop that movement. Okay, let me summarize. The olive input to the cerebellum reports not just the visual target, but also direction of the planned movement. Visual information is transmitted to type 1 cells, movements to type 2 cells. Simple spikes of type 1 cells accelerate the movement. Simple spikes of type 2 cells decelerate the movement. Okay. So what? Who cares? Why do I ask this question? This is my mom. And she lives down the street from me. On Wednesdays, I go to visit her. And, you know, we talk about things, how are things going in the lab and so forth. And, you know, I tell her. And so, you know, she would tell me, you know, we just started with this patient here who happens to live in Cleveland. So suppose he was here listening to your talk. What the heck would he say? He would say, who cares? How does this help me? That's great, Reza. You know, you publish your papers in these fine journals. You have these wonderful talks. But how does this provide anything useful to me? So I've been thinking about this, and so I want to show you the direction that I think this field, I would like it to go, and the way I would like to answer some of these questions. All right, so here's where we started from. We started from this 3-layer network. We said there's this mossy fiber input, there's these computations that are taking place, and what I've been telling you is things about what this computation might look like. Now, if you have a disease of the cerebellum, more than likely these Purkinje cells are going to die. And so you can sort of ask yourself, at least my mom would ask me, well, if you really think you understand the computation that's taking place here, you know, maybe you can build a machine that can take this input here and just bypass the circuitry here and produce the output that you need. Right? I mean, if you're man enough, right, put your money down and go and show me. It's not just in theory, but you can do that. So, you know, in order to do this, And this stuff, this is coming. This is just, I'm not saying anything new to you. This is the future of neuroscience, to replace things that are not working with machines. But that requires us understanding, what is actually supposed to be happening here in order for this machine to actually properly function. So what might that be? All right. So this is my student Amin, and what I'm going to show you is that the first step is you have to understand what is the intention for this movement here. So, I can't just constantly stimulate something. What I need to understand is this is the movement that I'm trying to do. That's the rest of the brain. Input to the cerebellum, mossy fibers. And then once you understand what the brain is trying to do, the rest of the brain is trying to do, maybe you can intervene and produce the correct output from the cerebellum. The first step is you have to understand the input to the cerebellum, which means decode the mossy fibers that are providing information from the rest of the brain to the cerebellum, saying, this is what I'm trying to do. You now tell me how well to do it. So let's record from the mossy fibers and understand the input that's coming when a saccade is about to take place. And this is what it looks like. So we can record from mossy fibers. And again, this is one of the beautiful things about the fabulous silicon probes that are now available. You can record wholesale from a large number of mossy fibers at the same time as you're recording from the Purkinje cells and the molecular layer interneurons in the cerebellum. What does it look like? Well, here's one mossy fiber. Let me show you what it looks like. So each one of these dots, of course, is a spike. And what Amin has done is that he's plotted for you the response of this mossy fiber. So this mossy fiber is coming from some input in the pons and is providing information about something else in the brain. And if you look at it, it kind of looks like the superior colliculus. It has a region of activity in, say, about 3 to 4 degrees. It has this mound, and it is telling you where the goal of this movement is going to be. So you have a group of Purkinje cells that we have a group of mossy fibers that when we decode, we can understand the goal of the movement in real time. Before the movement takes place, there's activity in the colliculus that's being sent via the pons to the cerebellum, and you can see it in the mossy fibers, and they look like this. There's another group of cells that are providing input, and we think these are the burst generators because their activity looks like muscles, or the burst generators that you would expect before they go to the motor neurons. So you see their activity basically grows bigger and bigger the greater the velocity of the saccade, the greater the amplitude of the saccade, just like you would expect if you look at the classic work of David Robinson and others that shows you basically this is what you get from burst generators. So this input looks like a copy of the motor commands in motor coordinates. All right. There are other things that come to the cerebellum, but let's focus on these two basic inputs. So what I'm showing you is that the mossy fibers to the cerebellum are providing you with two critical pieces of information. The first piece is where I'm supposed to go. It's a map of the colliculus space that's coming, that's saying this is where the goal is. There's a second input that's providing you input regarding the motor commands that are being generated in real time that's being sent to the cerebellum. Okay, now, Lance Optican, a couple of decades ago, suggested that in principle, maybe what the cerebellum is doing is as follows. It's integrating these commands to estimate in real time where you are with respect to where you were supposed to be. This is the estimated sensory state. This is the desired state. Compare those two. When you reach that boundary, stop your movement. So the idea is that you are integrating one kind of input to a bound set by another input, and the fundamental computation that's taking place is an integration to a bound. When you reach it, you stop the movement. Now, if you look at this circuit, what you see is something really interesting. This is going from motor coordinates to sensory coordinates. This is called a forward model. So those three ideas that I showed you at the beginning, dysmetria, learning, forward models, I think they all come together in this hypothetical, I don't know if this is true, is hypothetical description of what the computations in the cerebellum might be, which is an integration to a bound. But that integration is going from motor coordinates to sensory coordinates. And so who knows if this is true or not, but this is the outline of the computation that I think is taking place. And if one were to build this, neurostimulator, you would do it by decoding in real time the goal and the motor commands in the mossy fibers. You would integrate and you would output a mechanism that would say, stop the movement now. So who knows? In the next 10 to 15 years, we'll see if we can actually achieve something like this. All right. So what's the hole? And there are many holes in this story that I've been telling you. Let me go over some of the holes for you. Why is the olive reporting planned voluntary movements to the cerebellum? Because remember at the beginning I said that the olive was going to be telling you information about error. But, sure, a stimulus that appears at random is an unpredicted thing. I don't know where it was. So therefore that can be constituted as an error. But I also showed you that it's telling me where the movement is going. You saw that ramping activity before voluntary movement. So that's really odd. Why is the olive, that's not an error. It's an activity that's being sent to the cerebellum that doesn't match the concept of error. And I don't know the answer to that. Solomon is working on that. And I think it has something to do with the concept that olivary activity isn't just about learning, but it may be important for maintaining what it is that you've learned. So if you want your simple spikes to, for the rest of your life, to be modulated like I showed you, you need to have complex spikes that for the rest of your life will maintain that modulation, perhaps. Meaning that complex spikes cause learning, but they also produce sustaining of that memory. This is an idea which we perhaps can test. The second hole in my story is that how might these neurons that I've been describing to you perform this concept of integration to bound? How do you do integration of one signal to predict the sensory consequence and then compare it to another signal? Unfortunately, Amin has been making progress on this, and perhaps we will be able to map out the neural circuitry in the cerebellum and ask whether it performs a computation like this so that the Purkinje cells produce the output like I've been showing you. Another major hole is that, the output of the cerebellum is not Purkinje cells. It's the nucleus neurons. And if you've been paying attention to today's talk, you realize I didn't tell you anything about the nucleus neuron. Why? Because I don't understand it. I don't know how to make sense of the neurons there. I don't have a coordinate system for it. And if you trust me, it's not just me who doesn't understand it. We just don't know how to make sense of the output of the cerebellum yet. And that really remains to be seen. Alden has been working on this problem. The final thing that I want to show you is that you might say, well, Reza, saccades are such simple behavior. It's just an eyeball. Perhaps there's something special about that. I study reaching movements. I study leg movements. Does this help me at all? And I've been worried about that as well. And so I've been studying this movement alongside of eye movements. And it's a really cool movement that my marmosets perform. They are exceptional controllers of their tongue. They use it for getting things out of trees, and they vocalize extremely well. They are highly vocal animals. So Paul has been looking at cerebellar control of this beautiful movement that I'm showing you. And I, basically I want to know, do these ideas, this concept of organizing things based on climbing fiber activity, tell me anything about control of movements in general? Is there something special about saccades? And we'll see. We'll see how that story turns out. Okay, so let me finish. The main idea. Imagine the cerebellum as a school. Every classroom has a teacher, that's the climbing fiber input, and we're going to organize the Purkinje cells based on who's their teacher. When we do that, what we see that the simple spikes are suppressed during fixation. I showed you that act of holding still requires the cerebellum. The simple spikes predict timing and magnitude of when to stop that movement, and they increase their synchrony during saccades. And we think that this is coming about via something like an integration to bound that the cerebellum is getting input that says, here's where I'm trying to go. Here are the commands that I'm generating. You compare where I'm gone to where I'm supposed to go. And when I get there, stop my movement. I'm going to end with a poem with Rumi, 13th century Persian poet, and he goes like this. He says, every storm the beloved unfurls permits the sea to scatter pearls. Thank you for your time. Happy to answer your questions.
Questions for Dr. Shadmehr?
Yes, sir.
So the target-directed saccades, what happens if the monkey makes an error? Are these all just anticipation signals, or do they reflect an error?
I explain to me one more.
So if you have a target and the monkey is supposed to make a saccade in that direction,
yeah.
What if the monkey makes a mistake and makes a saccade in another direction?
If it makes a movement in another direction, we've only interpreted that as being a voluntary act, not as a mistake. We do measure movements and see that there are small errors at the end of them. So they're typically not like a 45-degree directional effect, but like a half a degree at the end and so forth. That reflects these errors that I showed you. You don't have to jump the target. You can just look at normal movements and you can see the endpoint errors that they have, and they respond very much the same as what I've shown you. Is that along the way you've been thinking?
Yeah, I mean, I'm sort of relating to the delayed response task where you've got a delay cell that predicts whether that's you're going to have this integration or this persistence.
Yeah, the idea would be that you would have a separation between where the target is and where I intend to move. And at least in terms of complex spikes, we see a separation between those two things. And this has been one of the nice things about saccades is that it's such a rich data set. The animal makes thousands of these movements in the course of a 3-hour recording, and you can't help but get conditions that provide you with this. Yes, ma'am.
Maybe a similar question. So it seems like a lot of this analysis is based on the idea of looking at the times in which the animal chose not to saccade to the target.
Yeah.
Do you think there's anything different? Thank you. Do you think there's anything different in the state of the system when they're essentially making a choice not to fixate on the target?
So we trained conditions for the animals so that they were rewarded regardless of whether they made a saccade to our target or not. So we had conditions where we said, all right, now we're going to reward you for any saccade. We're going to reward you for making a saccade to this target. And in that scenario, the animal has an option of whether they wish to make a saccade to the target or not. And that provided us then with the richness of the data that I showed you. Now, if you wanted to rely on just errors, meaning that they simply decide for whatever reason, maybe they got frustrated or something like that, I think that would not probably, it would not give us probably a whole lot of data, because these are scenarios where it's a dimly lit room, there's only one thing on the screen. It's going to be pretty hard for them not to pay attention to that and not to make a saccade. So in these cases, we purposefully gave the animal the option of making a movement to the target or not.
So you don't think there's any differences in like the motivation or the reward, like for how they were approaching that problem?
The motivational effects you can see on two things: pupil size and the velocity of the saccade, the vigor that they make it. And just like one of the interesting things about these animals is that the way we train them is to have them perform saccades for food. So they make a saccade and they get a little bit of food. But we made it so that the licking was a difficult thing. As you saw, the tube is placed so that it is hard for the animal to get their tongue in. So what they naturally do is this interesting foraging behavior. They work. As they work, food accumulates, then they go and lick. So one of the things we see is this trade-off between how long to work in order to go get food. And so you can make them highly motivated by making them easy to get their food. The tube is close to their mouth, or you put the tube farther away, it gets harder for them to get food. And they're less motivated, but what's interesting about that is that they work longer so that they get more food, and then they go and they lick it. So there's a component of decision making that you see in their behavior. It's just such highly intelligent animals. Then there is, you can make them motivated or less motivated by making how effortful it is to get their reward, and they will do the task for you. And there's another aspect of this which has to do with this interplay between their expectation of effort it takes to get reward, how many trials should they do before they go and harvest the reward. That is this other aspect of behavior that comes along for the ride.
If you induce an error, by giving them, let's say, for lack of a better word, you give them a gun that's off, it shoots off to the right. Can you watch the recalibration? Right.
So it induces plasticity. And a line of the experiments is to persistently give them error in the same direction. And then you see that the eyes now begin to adapt. So instead of moving to the target, now they begin to reduce that error that you had produced at the end of the movement. And that's that, you see that plasticity, yeah.
Anybody know any good songs?
Actually, I was going to ask a question.
Yes.
You touched on it a little bit, but are there analogous circuits or systems for limb movement?
Right. So one aspect of the work maps on nicely to a puzzling result that people had regarding arm movements. So difficulty, with arm movements is that the coordinate system is really difficult to understand. We don't know it in the motor cortex, and we don't know it in the cerebellum. The success that we've had is because really other people have figured out the coordinate system on the superior colliculus, and we just borrowed essentially that concept. Now, for tongue movements, what we've done is to do the following. If it's true that complex spikes suppress the simple spikes, perturb the system briefly, then the downstream effect is a small deflection that comes when there is a complex spike. So if you align behavior to when a complex spike occurred, you might be able to see the directional effect of this Purkinje cell. It's basically spike-triggered averaging, like you would do if you were doing it from the motor cortex perhaps, but now you're doing it via the climbing fiber and the Purkinje cell. We hope that by understanding the coordinate system of the climbing fiber input, in this case for tongue movements, we'd be able to understand its CS on, and then organize the simple spikes based on CS on to see if the same idea bears fruit.
Any other questions? Let's thank our speaker then.
Thank you so much.